Hi, Nicole Mashburn. Today we're going to talk about the resting membrane potential. So you have learned something about a neuron, and neurons are the cells that the nervous system uses to send signals to other parts of the body. And the signals that they use are called electrical impulses, or also called action potentials. And in order for a neuron to do this, they have to be set up in advance. They're special cells that are able to do this, and that's because they have something called a resting membrane potential, which essentially means they have a charge. Neurons are sitting there at rest with a charge on them. And if you could take a voltmeter and measure the charge on that cell, you would find that it's minus 70 millivolts in neurons, where the inside of the cell is negative compared to the outside of the cell. And the way this happens is you have a difference in the ionic makeup, or remember sodium and potassium chloride, the different ions. There's a different amount on the outside of the cell than the inside. So your intracellular fluid and your extracellular fluid have different amounts of sodium and potassium. And also the membrane is differentially permeable. It allows sodium and potassium to cross the membrane at different rates, and this causes there to be a charge on the membrane. So if you think back to earlier units, we talked about ion distribution in your cells and in your body. Well, if you're thinking about a neuron, in the extracellular fluid, the fluid that bathes the cells, there's more sodium relative to the amount of sodium inside the cell, and there's more potassium inside the cell relative to the amount of potassium outside the cell. So these are your positively charged ions, and you have some negatively charged chloride on the outside, which balances out with the negatively charged proteins on the inside. So you want your charges to balance, okay? But in the neuron, that doesn't happen. You don't have the equal number of pluses on the inside and the outside, so there's no balance. There's something going on that causes the inside to be negative relative to the outside. And this looks like the plus and minus of a battery. That's exactly what's going on. You're essentially setting up a battery that has the capability to send an electrical impulse. How does that happen? If you look at a neuron, you have differences in membrane permeability. And you have leak channels, and think back to your earlier discussion on how things move across a membrane. Remember, you have proteins that are inserted in the membrane, and some of them are just simply channels that allow things to move through them. They don't have to open or close. Things can just leak down their concentration gradient. So think about diffusion, where things go from a high level of concentration to a low level of concentration. So for example, if you had a beaker of water and you dropped a red food coloring into that beaker, over time the red would diffuse from that high concentration from that drop throughout the water to the low concentration. So you have these leak channels that allow sodium and potassium to move from the high level to the low level. So for example, if you had a leak channel for potassium, potassium's higher on the inside than it is the outside, it's going to diffuse out of the cell. Well, you have 75 leak channels of potassium compared to 1 of sodium. So if you have all these leak channels for potassium and all this potassium is going out, so let's say 75 potassiums go out for every 1 leak channel that allows sodium to go in, because sodium would go from high to low, it would go inside the cell. Well, look at your charges. So if all these K charges, these potassium charges, if you have 75 pluses leaving the cell and only 1 sodium plus entering the cell, then you're going to become more negative on the inside. So you're leaving the inside of the cell negative relative to the outside. So you've basically charged the battery. You've set up a resting membrane potential. There's actually a charge difference between the inside and the outside. You don't really have to worry about what's going on with the proteins. They don't move, and chloride goes back and forth freely, and so the negatives balance out. So you're just worried about the positives. Now, if you allowed this to happen over time, eventually these cells would reach equilibrium. Potassium would continue to leave the cell until finally it was equal on both sides. Well, if that happened, you wouldn't have a battery anymore. So you have a special pump called the sodium potassium pump, which uses ATP, which means it uses energy. So it is a active transport. If you go back to Unit 1 and think about passive transport versus active transport, it uses energy to pump potassium against the concentration gradient back into the cell and to pump sodium against its concentration out of the cell. So this helps maintain this high amount of potassium in the cell and high amounts or low amounts of potassium outside the cell, with your higher sodium outside the cell and lower sodium inside, because of this sodium potassium pump. So having a disproportionate amount of potassium link channels compared to sodium channels, and by having this pump, allows your neurons to have a resting membrane potential where the inside of the cell is negative relative to the outside. So it's essentially a battery. Think about a Duracell having a positive and a negative end. You've essentially built a battery. So this is a figure of your book that kind of walks you through the generation and maintenance of a resting membrane potential and talks to you about the different types of channels and how sodium potassium are able to move. I know some people like to read the chapter and they want to read these figures, but I've actually inserted an animation which I think makes a little more sense. So for you guys that like to read, here is a slide with all that on there, but I would implore you to watch this next short clip and really understand and see how the resting membrane potential is generated and maintained. The next time you see me, we'll be talking about action potentials. Thank you.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.